Chapter 6: Anatomy of flowering plants. You can very easily see the structural similarities and variations in the external morphology of the larger living organisms of both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have cells as the basic unit. Cells are organized into tissues, and in turn the tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations to diverse environments. 6.1. The tissues. A tissue is a group of cells having a common origin and usually performing a common function. A plant is made up of different kinds of tissues. The tissues are classified into two main groups, namely meristematic and permanent tissue, based on whether the cells being formed are capable of dividing or not. 6.1.1. Meristematic tissues. Growth in plants is largely restricted to specialized regions of active cell division called meristems. Greek meristos divided. Plants have different kinds of meristems. The meristems which occur at the tips of roots and shoots and produce a primary tissues are called apical meristems. Root apical meristem occupies the tip of a root, while the shoot apical meristem occupies the distant most region of the stem axis. During the formation of leaves and the elongation of stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in the axils of leaves and are capable of forming a branch or a flower. The meristem which occurs between mature tissue is known as intercalary meristem. They occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristem and intercalary meristem are primary meristems because they appear early in life of a plant and contribute to the formation of the primary plant body. Figure 6.1. Apical meristem. A. Root. Central cylinder. Cortex. Protoderm. Initials of central cylinder and cortex. Initials of root cap. Root cap. Root apical meristem. D. Shoot. Leaf. Primordium. Shoot apical meristematic zone. Axillary bud. Differentiating vascular tissue. A meristem that occurs in the mature regions of root and shoot of many plants, particularly those that produce woody axils and appear later than primary meristem, is called the secondary or lateral meristem. They are cylindrical meristems, fascicular, vascular cambium, interfascicular cambium, and cork cambium are examples of lateral meristems. These are responsible for producing the secondary tissues following division of cells in both primary and as well as secondary meristems. The newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cells and constitute the permanent tissues. During the formation of the primary plant body, the specific regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues. 6.1.2. Permanent tissues. The cells of the permanent tissues do not generally divide further. Permanent tissues having all cells similar in structure and function are called simple tissues. Permanent tissues having many different types of cells are called complex tissues. 6.1.2.1. Simple tissues. A simple tissue is made up of only one type of cells. The very simple tissues in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma forms the major component within the organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have small intercellular spaces. The parenchyma performs various functions like photosynthesis, storage, secretion. The collenchyma occurs in layers below the epidermis in most of the dicotyledonous plants. It is found either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to the deposition of cellulose, hemicellulose, and pectin. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant, such as young stem and petiole of a leaf. Sclerenchyma consists of long or narrow cells with a thick and lignified cell wall having a few or numerous pits. They are usually dead without protoplast. On the basis of variation in form, structure, origin, and development, sclerenchyma may be either fibers or scleroids. The fibers are thick-walled, elongated, and pointed cells, generally occurring in grooves in various parts of the plant. The scleroids are spherical, oval or cylindrical, highly thickened dead cells with very narrow cavities, lumen. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear, and sapota, seed cores of legumes, and leaves of tea. Sclerenchyma provides mechanical support to the organs. Figure 6.2 Simple tissues. A. Parenchyma with intercellular spaces. B. Collenchyma with thickened corners, protoplasm, vacuole, cell wall. C. Sclerenchyma, a fiber, lumen, thick cell wall. A scleroid, pits, lumen, thick cell wall. 6.1.2.2 Complex tissues. The complex tissues are made up of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissues in plants. Figure 6.3 A. Xylem, tracheid, vessels. B. Phloem with sieve pore, sieve tube element. Phloem, parenchyma, companion cell. Xylem functions as a conducting tissue for water and minerals from roots to the stem and leaves. It also provides mechanical strength to the plant parts. It is composed of 4 different kinds of elements, namely tracheids, vessels, xylem fibers, and xylem parenchyma. Gymnosperms lack vessels in their xylem. Tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layers of the cell walls have thickenings which vary in form. In flowering plants, tracheids and vessels are the main water transporting elements. Vessel is a long cylindrical tube-like structure made up of many vessels, called vessel members, each with lignified walls and a larger central cavity. The vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforations in their common walls. The presence of vessels is the characteristic feature of angiosperms. Xylem fibers have highly thickened walls and obliterated central lumens. These may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell walls are made up of cellulose. They store food materials in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by the ray parenchymatous cells. Primary xylem is of two types, protoxylem and metaxylem. The first formed primary xylem is called metaxylem. The first formed primary xylem elements are protoxylem and the later formed primary xylem is the metaxylem. In stems, the protoxylem lies towards the center pith and the metaxylem lies towards the periphery of the origin. This type of primary xylem is called endarch. In roots, the protoxylem lies towards periphery and the metaxylem lies towards the center. Such arrangement is primary xylem. Such arrangement of primary xylem is called exarch. Phloem transport food materials usually from leaves to other parts of the plant. Phloem in angiosperms is composed of sieve tube elements, companion cells, phloem parenchyma, and phloem fibers. Gymnosperms have albuminous cells and sieve cells. They lack sieve tubes and companion cells. Sieve tube elements are also long tube-like structures arranged longitudinally and are associated with the companion cells. Their end walls are perforated in the sieve-like manner to form the sieve plates. The mature sieve elements possess a peripheral cytoplasm and a large vacuole but lacks a nucleus. The functions of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The sieve tube elements and companion cells are connected by epithelials present between the common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated tapering cylindrical ends which have a dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmadesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fibers, bast fibers, are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are found in the secondary phloem. These are much elongated and branched and have a pointed needle-like apices. The cell wall of phloem fiber is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute and flax and hemp are used commercially. The first formed primary phloem consists of a narrow sieve tube and is referred to as protophlloem, and the later formed phloem has bigger sieve tubes and is referred to as metaphloem. 6.2 The tissue system. We were discussing types of tissues based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. The structure and function would also be dependent on the location. On the basis of their structure and location, there are three types of tissue system. These are the epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. 6.2.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form continuous layer. Epidermis is usually single-layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall with a large vacuole. The outside of the epidermis is often covered with a waxy, thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in the roots. Stomata are structures present in the epidermis of the leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two beanie-shaped cells known as guard cells, which encloses the stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer walls of guard cells away from the stomatal pore are thin, and the inner walls toward the stomatal pore are highly thickened. Guard cells possess chloroplast and regulate the openings and closing of the stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as the subsidiary cells. The stomatal aperture guard cells and the surrounding subsidiary cells are called the stomatal apparatus. Figure 6.4. Diagrammatic representation of A. Stomata with bean-shaped guard cells, epidermal cells, subsidiary cells, chloroplast, guard cells, and stomatal pore. B. Stomata with dumbbell-shaped guard cell, epidermal cell, subsidiary cells, guard cells, and no chloroplast and stomatal pore. The cells of the epidermis bear a number of hairs. Root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system is usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to the transpiration. 6.2.2 The ground tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in the cortex, pericycle, pith, and medullary veins in the primary stem and roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. 6.2.3 The vascular tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and the phloem together constitute vascular bundles. Figure 6.5. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues, and hence are called open vascular bundles. In monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoined type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoined vascular bundles usually have the phloem located only on the outer side of the xylem. Figure 6.5 Various types of vascular bundles. A. Radial with xylem and phloem. B. Conjoined closed phloem xylem. C. Conjoined open phloem cambium then xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots, stems, and leaves, it is convenient to study the transverse section of the mature zones of these organs. 6.3.1 Dicotyledonous roots. Look at figure 6.6a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root heads. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as the radial walls of the endodermal cells have a deposition of outer A winter impermeable waxy material. Suberin in the form of Casparian strips. Next to the endodermis lies a few layers of thick walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, the cambium ring develops between the xylem and phloem. All tissues on the inner side of the epidermis, such as pericycle, vascular bundles, and pith, constitute the stele. Figure 6.6. Transverse section of dicot root, primary, with root hair, epidermis, cortex, endodermis, pericycle, protoxylem, metaxylem, pith, phloem. B. Monocot root with root hair, epidermis, cortex, endodermis, pericycle, phloem, protoxylem, metaxylem, pith. 6.3.2 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. Figure 6.6b. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles. In the monocot root, pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. 6.3.2 to 6.3.3 Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may bear trichomes and a few stomata. The cells arranged in the multi-layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of parenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of around thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as the starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of dicot stem. Each vascular bundle is conjoined, open, and with exarch protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. 6.3.4 Monocotyledonous stem. Monocotyledonous stem has a sclerenchymatous hypodermis. A large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and the water containing cavities are present within the vascular bundles. Figure 6.7 Transverse section of stem. A. Dicot with epidermal hair, epidermis, hypodermis, parenchyma, endodermis, pericycle, vascular bundle, medullary rays, pith. A. Epidermis, collenchyma, parenchyma, endodermis, pericycle, phloem, cambium, metaxylem, protoxylem, pith. B. Monocot with epidermis, hypodermis, vascular bundles, ground tissue, epidermis, hypodermis, vascular bundle, phloem, xylem, ground tissue. 6.3.5 Dorsiventral dicotyledonous leaf. The vertical section of a dorsiventral leaf through the lamina shows the 3 main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface, the axial epidermis, and lower surface, the abaxial epidermis, even the latter may even lack stomata. The epidermis, which covers both the upper surface, the axial epidermis and lower surface abaxial epidermis of the leaf has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The later may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possesses chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The axially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other, oval or round and loosely arranged. Spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at figure 6.8a and find the position of xylem in the vascular bundle. Transverse section of leaf A dicot with bundle sheath, xylem, phloem, adaxial epidermis, palisade mesophyll, air cavity, spongy mesophyll, substomatal cavity, stoma, abaxial epidermis. Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surface of the epidermis and the mesophyll is not differentiated into palisade and spongy parenchyma. Figure 6.8b. In grasses or an adaxial epidermal cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to the water stress, the leaves curl inwards to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles, except in main veins, as seen in vertical sections of the growth. Transverse section of leaf, B monocot, adaxial epidermis, xylem, mesophyll, substomatal cavity, abaxial epidermis, stoma, phloem. 6.4 secondary growth. The growth of the roots and stems in the length with the help of apical meristem is called the primary growth. Apart from primary growth, most dicotyledonous plants exhibit an increase in growth. This increase is called the secondary growth. The tissue involved in secondary growth and are the two lateral meristems, the vascular cambium and the cork cambium. 6.4.1 Vascular cambium, the meristematic layer that is responsible for cutting of vascular tissues or xylem and phloem is called the vascular cambium, and the young stem is present in patches as a single layer between the xylem and phloem. Later it forms a complete ring. 6.4.1.1 Formation of cambial ring. In dicot stems, the cells of cambium present between primary xylem and the primary phloem is the intrafascicular cambium. The cells of medullary rays adjoining these intrafascicular cambium become meristematic and form the interfascicular cambium. Thus, a continuous ring of cambium is formed. 6.4.1.2 Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner and the outer sides. The cells cut off towards pith and mature into the seed secondary xylem, and the cells cut off towards periphery mature into secondary phloem. The cambium is generally more active on the inner side than on the outer. As a result, the amount of secondary xylem produces is more than the secondary phloem and soon forms a compact mass. The primary and the secondary phloems get gradually crushed due to the continued formation and accumulation of secondary xylem. The primary xylem, however, remains more or less intact in or around the center. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial directions. These are the secondary medullary rays. Figure 6.9 Secondary growth in dicot stem diagrammatic stages in transverse view. Epidermis, cortex, primary phloem, vascular cambium, primary xylem, pith, interfascicular cambium. Medullary rays, secondary xylem, secondary phloem, cambium ring, phloem, phloem. 6.4.1.3 Springboard and Autumn board. The activity of cambium is under the control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform throughout the year. In the spring season, cambium is very active and produces a large number of xylem elements having vessels with wider cavities. The board formed during the season is called the spring board or early board. In winter, the cambium is less active and forms fewer xylem elements that have a narrow vessels, and this board is called the autumn board or late board. The spring board is lighter in color and has a lower density, whereas the wood formed during the season is called the spring wood or early wood. In winter, the cambium is less active and forms few salary elements that have narrow vessels, and this wood is called the autumn wood or late wood. The spring wood is lighter in color and has a lower density, whereas the autumn wood is darker and has a higher density. The two kinds of wood that appear in an alternate concentric rings constitute an annual ring. Annual rings seen in a cut stem give an estimate of the age of the tree. 6.4.1.4 Heartwood and sapwood. In older trees, the greater part of the secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layer of the stem. These substances make it hard, durable, and resistant to the attacks of microorganisms and insects. This region comprises dead elements with highly lignified walls and is called heartwood. The heartwood does not conduct water, but it gives a mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as the sapwood. It is involved in the condition of water and minerals from root to leaf. 6.4.2 Cork cambium. As the stem continues to increase in girth due to the activity of the vascular cambium, the outer cortical and epidermis layers get broken and need to be replaced to provide a new protective cell layers. Hence, sooner or later, another meristematic tissue called cork cambium or phellogen develops usually in the cortex region. Phellogen is a couple of layers thick. It is made up of narrow, thin-walled and nearly rectangular cells. Phellogen cuts off cells on both sides. The outermost cell differentiate into cork or phellem, while the inner cells differentiate into secondary cortex or phelloderm. The cortex is impervious to water due to the suberant deposition in the cell wall. The cells of secondary cortex are parenchymatous. Phellogen, phellem, and phelloderm are collectively known as the periderm. Due to the activity of the cork cambium, pressure builds up on the remaining layers peripheral to the phellogen, and ultimately these layers die and slough off. Bark is a non-technical term that refers to all tissues exterior to the vascular cambium, therefore including secondary phloem. Bark refers to a number of tissue types, which is periderm and secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. Name the various kinds of cell layers which constitute the bark. At certain regions, the phellogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cells. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped openings called lenticels. Lenticels permit the exchange of gases between the outer atmosphere and the internal tissue of the stem. These occur in most woody trees. Figure 6.10. A. Lenticel with epidermis, complementary cell, cork cambium, secondary cortex. B. Bark. 6.4.3 Secondary growth in roots. In the dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem, bundles a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular. Further events are similar to those described above for a dicotyledonous stem. Secondary growth also occurs in stems and pools of gymnosperms. However, secondary growth does not occur in monocotyledons. Figure 6.11. Different stages of secondary growth in a typical dicot root. Epidermis, cortex, phloem, primary phloem, cambial ring, endodermis, pericycle, protoxylem, epidermis, cortex, vascular cambium, secondary phloem, primary xylem, secondary xylem, cortex, epidermis or periderm, cortex, primary phloem, annual ring, secondary xylem, secondary phloem raised, vascular cambium, secondary phloem, primary xylem, secondary xylem. Summary: anatomically, a plant is made up of different kinds of tissues. The plant tissues are broadly classified into meristematic, apical, lateral, and intercalary, and permanent, simple, and complex. Assimilation of food and its storage, transportation of water, minerals, and photosynthesis, and mechanical support are the main functions of tissues. There are three types of tissue system: epidermal, ground, and vascular. The epidermal tissue systems are made up of epidermal cells, stomata, and the epidermal appendages. The ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle, and pith. The vascular tissue system is formed by the xylem and phloem on the basis of presence of cambium. Location of xylem and phloem. The vascular bundles are of different types. The vascular bundles form the conducting tissue and translocate water, minerals, and food material. Monocotyledonous and dicotyledonous plants show marked variation in their internal structure, the different type, number, and location of vascular bundles. The secondary growth occurs in most of the dicotyledonous roots, stem, and leaves. Roots and stems, and it increases the girth diameter of the organs by the activity of the vascular cambium and the cork cambium. The wood is actually a secondary xylem. There are different types of wood on the basis of their composition and time of production.